Imagine a world where scientists could unlock the secrets of life itself, altering the building blocks of life, the DNA, to solve some of humanity's biggest challenges. This isn't just science fiction, it's the reality of recombinant DNA technology, a tool that has changed the landscape of science, medicine, and agriculture. Why is this technology so important? Think about the breakthroughs it has brought. It enables the production of vital drugs like insulin, helping millions of diabetic manage their condition. It also paves the way for pest-resistant crops, ensuring food security for a growing global population. Even more, it helps researchers study genetic diseases, opening doors to potential treatments and cures. The process of recombinant DNA technology, the steps involved in recombinant DNA technology are fascinating, each one contributing to the creation of a powerful genetic tool.
First is isolating the DNA. The process begins by isolating the DNA containing the gene of interest. For example, imagine scientists identifying and extracting the insulin gene from a human cell. This involves breaking open the cells to release their contents and separating the DNA from other cellular components. A specific gene, such as the one responsible for insulin production, is located using advanced techniques like gene probes.
Secondly, cutting the DNA. Once the DNA is isolated, it's time to cut it into precise segments. This is done using restriction enzymes, which act like molecular scissors. These enzymes recognizes specific sequences of DNA and make precise cuts in those sites. For example, the restriction enzymes accurately cuts DNA at a sequence called GAATTC. This ensures that the gene of interest is nearly separated and ready for the next step. In the third steps, creating a recombinant vector. Next, the gene of interest is inserted into a vector, a carrier molecule that can transport foreign DNA into a host organism. Plasmid, small circular DNA molecules found naturally in bacteria, are commonly used as vectors. The plasmid is first opened using the same restriction enzymes to create a compatible ends. Then, the gene is inserted into the plasmid, and the two pieces are joined using an enzyme called DNA ligase, which act like glue to seal the DNA fragments together. The result is a recombinant plasmid, a vehicle that carries the desired gene. Fourth steps, introducing the vector into a host cell. The recombinant plasmid is then transferred into a host cell, such as a bacterium, through a process called transformation. This involves creating conditions that make the bacterial cell membrane more permeable, such as using heat shock or an electric pulse. The plasmid enters the bacterial cell, carrying the gene of interest with it. And the last steps, replicating and expressing the gene inside the host cell. The plasmid begins to replicate, producing multiple copies of itself. The bacteria divide rapidly, creating millions of cells that contains the recombinant DNA. But the real magic happens when the host cell starts expressing the gene, reading the genetic instructions to produce the desired protein. For example, if the inserted gene codes for insulin, the bacteria will manufacture human insulin, which can be harvested, purified, and used in medicine. The real-world application of recombinant DNA technology are astounding. It has revolutionized medicine by enabling the production of vaccines and treatments for diseases. In agriculture, it has helped create genetically modified crops like pest-resistant corn, ensuring better harvests. In research, it continues to break barriers, offering hope for therapies that can address genetic disorders.